Okay, this is the Professor, CBMD PhD, and today's Tuesday, July 11th, 1923. It is 2:35 and 19 seconds, a.m. Eastern Standard Time, and today's research, anti-cancer. So let's jump to it here. Immune and tumor cell, tug of war controls anti-cancer, activity, date July 5th, 1923. Source, St. Jude Children's Research Hospital. As scientists have found that altering amounts of nutrient glutamate in the tumor microenvironment can enhance or impair the immune system's anti-cancer response. If tumor cells use all of the available glutamate, then a specialized immune cell, type known as a dendritic cell, is starved of glutamate, leading to impaired anti-tumor immune function. If we can supplement enough glutamate to the tumor microenvironment, that will inhibit tumor growth, because dendritic cells will use it and activate the adaptive immune response. Dendritic cells turn on cancer-killing immune cells, called T cells. The group showed that reapplying glutamate in the tumor microenvironment severely reduced tumor growth, because dendritic cells were then better able to activate anti-cancer T cells. Famously, cancer cells secrete many signals to turn the immune response off in this area, especially the T cells that threaten their destruction. The research team is the first to identify a nutrient as a major signal between cancer cells and dendritic cells in the local environment. Immune checkpoint blockade therapy inhibits the off signals cancer cells send to immune cells that suppress the immune response in the tumor microenvironment. These therapies have been highly effective, but only in a small number of patients. The researchers found that supplying glutamate in a combination with checkpoint therapy enhanced anti-cancer activity in mice. The researchers provide a proof of concept that nutrients can act synergistically with checkpoint inhibitors for tumor treatment as a new strategy for combination therapy. While much cancer research has focused on T cells because of their direct cancer-killing activity, this study is one of the first to examine how the tumor microenvironment affects the dendritic cells, which activate T cells. The researchers found that without glutamate, dendritic cells failed to activate the T cells that directly kill cancer cells. Even though T cells are the cornerstone for anti-cancer immunity, they cannot do the job by themselves. We can think of dendritic cells as a driver and a T cell as the car. If you do not have a driver, the car will not move. Moreover, nutrients like glutamate serve as a license for the driver. And similarly, when the researchers remove the proteins that respond to or take up glutamate in dendritic cells, the immune cells fail to activate cancer-killing cells. These proteins, called FLCN and SLC3882, are important in sensing and acquiring nutrients, but have not previously been connected to immune cells' reactions to tumors. They may serve as potent drug targets to improve cancer therapy. Researchers are extremely excited about these findings. In addition to enhancing the therapeutic intervention, they have provided a conceptual advance by showing how nutrients mediate cell-cell communication, an understudied concept in the field of immunometabolism. And this is a professor of microbiology, bioinformatics, pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.